Welcome to the Explainer. Have you ever actually just stopped to watch a plant grow? Probably not, right? Because to our eyes, it happens in super slow motion. But what if I told you that beneath the surface of that quiet, still houseplant sitting on your desk is a raging, relentless engine of creation? Today, we are going to demystify the hidden mechanics behind the everyday miracle of plant life. We're going to uncover exactly how plants achieve something that we humans simply cannot do: the capacity for infinite growth. To put this incredible biological power into perspective, just think about this number: 17,500. Inside a single, everyday maize plant, the tiny, microscopic tip of its root, which we call the apical meristem, can produce over 17,500 new cells in just one single hour. I mean, that is an absolutely explosive rate of cellular generation, and it's happening silently, right under our feet in cornfields, literally everywhere. And wait, it gets crazier. It's not just about cranking out more cells, it's about how much those cells can expand. Take a watermelon, for instance. The cells inside a growing watermelon can increase in size by up to 350,000 times. It's a completely alien concept to us. When we reach adulthood, our bodies stop growing. We hit a limit. But plants, they possess what biologists call indeterminate growth. They just keep building, expanding, and reaching for literally as long as they live. Okay, let's get right into Section 1, the infinite growth engine. Okay, let's dive into this. Why on earth don't plants have a size limit like we do? What's their biological secret? Well, the answer lies in a very specific, specialized type of plant tissue called the meristem. Meristems are basically the biological engines that make this totally open form of growth possible. Think of them as localized factories placed at specific locations in the plant body. Like the tips of shoots reaching up for the sun and the tips of roots pushing down into the soil. The cells inside these meristems have one primary job, and that is to divide and self-perpetuate indefinitely. They're constantly adding brand new cells to the plant body, allowing it to literally outgrow its own maturity. Moving right along, that brings us to Section 2, measuring plant growth. So, we know this relentless biological expansion is happening constantly, but how do scientists actually measure it? I mean, we can't exactly put a plant on a bathroom scale to see how much it grew today in a meaningful biological sense. At a cellular level, growth is fundamentally an increase in protoplasm. That's the living substance inside a cell. But it's pretty darn difficult to measure protoplasm directly without destroying the cell itself. So instead, scientists have to rely on proportional physical parameters. Depending on the plant part, we might measure the increase in its fresh or dry weight. Or if we're looking at something like a pollen tube, we'll measure its length. For a broadleaf, we'd look at the increase in surface area. Or we might track volume. Or, as we learned with the maize root, the sheer increase in cell number. And as we measure this growth, we see that a plant cell goes through a seriously fascinating chronological journey. It's broken down into 3 distinct phases. First up is the meristematic phase at the very tip, where cells are rich in protoplasm and just constantly dividing. As new cells are born, the older ones get physically pushed further away from the tip into the second phase, elongation. Here the cells undergo increased vacuolation, they dramatically enlarge, and they lay down new cell walls. Finally, pushed even further back, they enter maturation. This is where they hit their maximal size and develop the specific structural modifications they need for their lifelong job. Now for my absolute favorite part, Section 3, the math of growth. Because here's the thing. As these cells divide and elongate, they don't just do it randomly. Plant growth isn't just some messy biological sprawl. It actually strictly adheres to beautiful mathematical models. The growth rate of a plant essentially takes one of 2 mathematical paths: arithmetic or geometric. In arithmetic growth, imagine a cell divides into 2. One daughter cell keeps the ability to divide, but the other one just stops and matures. This creates a steady, constant, linear rate of growth, kind of like a root elongating at a very steady pace. Geometric growth, on the other hand, is explosive. A cell divides into 2, and both of those new cells retain the ability to divide. So 2 become 4, 4 become 8. It creates a rapid exponential curve. And this brilliantly illustrates exactly what that looks like. You've got arithmetic growth looking like a linear, step-by-step addition. Geometric growth, however, just balloons outward exponentially. But obviously, in the real world, geometric growth eventually hits a wall. When nutrient supplies run low, that exponential spike slows down and levels off, creating a stationary phase. Out in nature, this forms a typical S-shaped curve, which is pretty much characteristic of all living organisms growing in a natural environment. To really wrap our heads around the efficiency of this growth, scientists use two different measurements. Absolute growth rate is simply the total growth per unit of time. Simple enough. But relative growth rate measures that growth compared to the plant's initial size. So imagine two leaves, a tiny leaf and a massive leaf. Over a week, both grow exactly 5 square centimeters. Their absolute growth rate is identical, right? But the tiny leaf essentially doubled its size, while the huge leaf barely changed at all. Therefore, the tiny leaf is working at a vastly higher relative growth rate. It's acting as a much more efficient engine of growth. Which leads us directly into Section 4, how plant cells specialize. So think about it. All these millions of new cells being rapidly produced are basically just blank slates at first. How do these newly minted cells figure out their specific career paths in the plant structure? Well, they figure it out through a process called differentiation. This is when cells undergo structural changes to perform highly specific functions, and they usually lose their ability to divide in the process. A perfect example is a tracheary element. That's a cell destined to become part of the plant's internal plumbing. To do its job effectively, this cell will intentionally lose its own protoplasm. It essentially hollows itself out completely and develops incredibly strong, elastic lignocellulosic walls. It sacrifices its ability to divide so it can act as a permanent, long-distance water pipe, even under extreme physical tension. But here is where plants show off a literal cellular superpower that we humans could only dream of: de-differentiation. Under certain conditions, living, mature cells that had already differentiated and completely lost their ability to divide can actually regain that capacity. It's kind of like a retired specialist being called back into action to become a general worker again. This happens when the plant desperately needs to form new meristems, like the court cambium, right from fully mature parenchyma cells. And no surprise, the cycle doesn't stop there. Once those de-differentiated cells start dividing and producing new tissue, those new cells will mature and specialize all over again, a process called redifferentiation. They mature, perform specific functions, and lose their dividing capacity once more. This whole cycle just goes to show the incredible open-ended flexibility of plant tissue generation. The destiny of a plant cell isn't a locked door. It's practically a revolving one. All right, on to Section 5, adapting through plasticity. Because they have this deep, open-ended cellular flexibility, plant growth isn't just following some rigid, unchanging script. It's incredibly adaptable to the environment and to the different phases of a plant's life. Which leads to a really fascinating question. Can a plant physically change its own shape to fit its environment or its age? Like, if you plant a seed in the water versus the soil, will the plant literally rewrite its own physical blueprints just to survive? The answer is absolutely yes. Plants are basically masters of morphological shape-shifting, which is an ability called plasticity. A really great example of this is heterophyly. In plants like cotton, coriander, or larkspur, the leaves of a juvenile plant are completely different in shape from the leaves of a mature plant. But it's not just about age, it's also about the environment. Take the buttercup plant. It will produce one specific shape of leaf if it grows in the air. Finally, we arrive at Section 6, plant growth regulators. So, who's the boss here? Who is coordinating this whole shebang? Who's telling the cells to divide exponentially, or to lose their protoplasm, or to completely change the shape of a leaf based on the water level? Meet the ultimate chemical managers of this entire intricate operation: plant growth regulators, or PGRs. Now, what's really interesting about this slide is how these chemical managers are broadly divided into two distinct teams, showing a beautiful division of labor. You've got the growth promoters like auxins, gibberellins, and cytokinins, and these are the gas pedals. They push hard for cell division, enlargement, and flowering. Then, on the other side, you have the stress responders and inhibitors like abscisic acid. These are the brakes. They help the plant survive wounds, deal with environmental stress, and manage dormancy. Ethylene, which is a gaseous PGR, can actually do a little bit of both, but it's largely known as an inhibitor. Together, these chemicals masterfully conduct the entire symphony of plant growth. Yet, for all their absolutely crucial importance, our discovery of these chemical managers was a total, absolute fluke. Seriously, the discovery of each of the 5 major groups of PGRs was entirely accidental. It actually started with none other than Charles Darwin and his son Francis. They were simply hanging out observing canary grass coleoptiles, which are those little protective sheets around the emerging shoot. They noticed the grass bending toward a light source, a process we now call phototropism. Their simple, totally accidental observation of grass bending toward a window literally sparked the entirely new science of plant hormones. So, as we wrap up the explainer today, I really want to leave you with this thought. Plants are not just passive green background objects decorating our world. The next time you look at a simple houseplant or stare up at a towering tree, ask yourself, what invisible geometric math is it calculating right now? What intense cellular specialization is it undertaking? What brilliant chemical engineering is it executing right in front of your eyes? Because beneath that quiet green surface, a masterful, infinite growth engine is always running. Thank you so much for joining me today, and keep questioning the world around you.